All right, let's go over folder 8. Which is about regulation of gene expression. Well, that term means DNA transcribed into the lowercase mRNA into protein. So this is transcription, as we said many times, as we said many times before, and translation. So this right there, that's what we mean by, still one more time, by that term, gene expression. Because remember, that term here is DNA. Any coding sequence, again, any coding sequence, I should define it, any DNA coding sequence is a gene. Because not every DNA sequence is, so again, not every DNA sequence is a gene, because not every DNA sequence is a coding sequence. Again, not every DNA sequence is a coding sequence. Now, the term coding sequence, when you say coding sequence, that means it's a sequence, that gets transcribed and then translated. So a sequence that gets transcribed and then translated is a coding sequence. And that sequence is said to be, still one more time, a coding sequence is a sequence that can be expressed. It's a sequence that has the information to make a protein. Sequence that has information to make proteins. What we need to do is see why making of proteins is regulated. Like, you don't make proteins 24/7 just because. So, and that's the topic being discussed in this folder. Again, regulation of the transcription and the translation of DNA coding sequences. Again, the regulation, the regulation of the transcription and translation of DNA coding sequences. All right. There is, of course, regulation in eukaryotes, and there is regulation in prokaryotes. We will limit our discussion to prokaryotes. So regulation of gene expression in prokaryotes. Once again, we will not discuss the eukaryotes. It's not going to be covered in this class. So regulation of gene expression only in prokaryotes. And to remind you again, bacteria, in bacteria. Now, as we said earlier, that the term gene expression is basically, I'm going to write this again, the DNA into the mRNA into protein Now we said this here is called transcription, right there, transcription, and this is translation. As I said it before, I'm just emphasizing that. Well, regulation can take place at this point, at this point, at this point, and at this point. So this is 1, this is 2, this is 3, this is 4. Now, so let's write down regulatory levels. 1, what's 1? Look where 1 is. 1 is messing with the transcription itself. So transcriptional control level, transcriptional control level. 2, 3, and 4. Look at 3 at least. 3 is messing with the translation. So this one is translational control level. 2 is after transcription, but before translation, but it's after transcription. So it's called post-transcriptional. Transcriptional control level. And of course, the number 4 is after translation, so post-translational control level. Now, since we are limiting our discussion right now, at least to bacteria or prokaryotes, you need to know that they only use 1 and 4. So once again, in prokaryotes, only 1 and 4 are used. So there is no post-transcriptional control in prokaryotes, and there is no translational control in prokaryotes either. It's only 1 and 4. Only 1 and 4. Why are we regulating anyway the making of proteins? Well, few reasons. I should not be making proteins unless the proteins are needed. Why? How come? Because if they are not needed now, let's say they are needed next week. So why are you making them now? Why? What's the issue of making them earlier? Well, you are limited space-wise. Remember, the cell is very tiny, so you have no storage area. So make them as needed. You cannot make them earlier because you have no room to store them anyway. And of course, do not make any proteins that are not needed whatsoever, because if you are making proteins that are not needed, you are wasting energy because you don't make any proteins for free. There is energy invested. So once again, you only make proteins as needed. And that's why the making of proteins must be regulated. All right. Now, since we are limiting our discussion, at least now, for bacteria or prokaryotes, remember again, we have only 1 and only 1 and 4. And these are the transcriptional and the post-translational. All right. The transcriptional control. Transcriptional control level. And one more time, look where we at, really here. We are regulating transcription. We are looking at 2 mechanisms. One of them is enzyme induction. And for the enzyme induction, we are using the LAC for the sugar lactose. Again, LAC for the sugar lactose. Operon, and we'll define that term soon. And we are also using enzyme repression. And for enzyme repression, we are using TRP for the amino acid tryptophan. Again, the amino acid tryptophan, and that same term again. All right. And that will take care of the transcription in bacteria or prokaryotes. So we want to investigate both enzyme induction and enzyme repression. The difference between the two is, I want to say, minor and major at the same time, as you will see. All right, let me take you back to a bacterial cell to show you where we will be. So assume this is a bacterium, and we are using right now the E. coli, the name of the bacterium, Escherichia coli. I said before that the chromosome is circular, again, circular, it's very folded and found in the nucleoid region. So let's say somewhere here, right there, let's say that's a nucleoid region, like this area. So this is where the chromosome will be, right? In that area, the nucleoid region. All right, so, and it is circular, but it's not just very, like a basic circle, it's very folded and twisted, right? So in theory, it's very twisted. So I said it's huge, but folded and coiled. And then you close that circle, right? And it is found in that nucleoid region. But what I want to do right now is actually simplify it and say it is just one basic unfolded circle. It's not the case. Let me actually erase that again and do a perfect circle. It is not the case, but we need to do so for simplicity right now. All right. So let's say that this here is the bacterial chromosome. Here, bacterial chromosome. And as you can tell, it's circular. Well, somewhere on that chromosome, let's say from here to here. So let's say this one right there is the lac operon, and I said we'll define that. And let's say somewhere else, let's say from here to here, for example, let's say this is the TRP for tryptophan, TRP for tryptophan operon. So I want you to know where we at when I start navigating through the system. So what I'm going to do next, because I want to look at enzyme induction first, enzyme induction first. So I'm using the lac first. So what I'm going to do, I'm going to take this sequence out. What sequence? This one here. So you see where it is in the bacterial chromosome. So let's say this is what it is right now here. This is this. That's where it is, a sequence on the bacterial chromosome. A sequence on that bacterial chromosome. So I'm taking it out so I can really examine it closely. So let's label that again. This here is my my lac, sorry, here, my lac. That term again that I will define. The concept, before I get into it, when you say enzyme induction, what does that mean? Like the term, that means lactose is a sugar. Lactose is a sugar. The bacterial cell will not make the enzymes that are needed to metabolize it unless it's there. Like, why will I make enzymes that needed to break down the sugar lactose if lactose is not present? So when you say enzyme induction, what does that mean? That means the presence of lactose. When lactose is present to the bacterial cells, lactose will induce the cell, hence the induction, induce the cell to make the enzymes that are needed to break down the sugar lactose. You only make a trip to the kitchen to grab a fork, a knife, to eat, let's say, a piece of steak, if someone is offering you that piece of steak. If I'm giving you like that juicy piece of steak to eat, but no fork, no, well, you have to make a trip, I guess, to the kitchen to go grab like something to eat that fork with. But if you, I'm sorry, something to eat that steak with. But if you have no food, why do you care to go to the kitchen to grab a fork? To do what with the fork? So that's the concept here for enzyme induction. I will repeat. Bacterial cells will only care to make the enzymes that are needed to break down the sugar lactose if lactose is present. If lactose is not present, it is silly to make enzymes that are only needed to break down lactose, but lactose is not even available. Like, that's a waste of time. Now, of course, enzymes, remember, keep in mind, they are proteins. As we keep saying, enzymes are proteins. So remember, at the regulation of gene expression, I care to make the proteins, or I care not to make the proteins at the present time. So we are deciding, should I make those proteins that are needed to break down lactose, or should I not make the proteins that are needed to break down lactose right now? All right. Now, for the enzyme repression, look at the difference. Tryptophan is an amino acid that bacteria cannot live without. Once again, tryptophan is an amino acid that bacteria cannot live without. So now, if tryptophan is not available, like the bacteria do not have tryptophan, well, guess what? They are not complaining. They are making the enzymes that are needed to make tryptophan, not to break it, to make tryptophan. The second you start supplying the bacteria with tryptophan yourself, you started artificially supplying tryptophan to the bacteria. Guess what you just did to them? You are repressing them from making the enzymes that are needed to make the tryptophan. Because why will they make it if you are giving it to them? So here I need to make tryptophan, I need to build it. Here I need to break lactose, because lactose is food. I need to break it down to use it for energy. And we'll say that example again as we proceed with enzyme induction, enzyme repression. Now, I said in the announcement about this specific concept is 2,000,000% conceptual. There is really nothing to memorize. It's conceptual. So let's go back to the Lac opera. It's a DNA sequence. Well, remember again, where did I get it from? I grabbed it from the chromosome. Where is that cell? Here. Where is that bacterial cell or bacterium? That's a chromosome. And look, I took that sequence out. That's a Lac. So it is a DNA sequence. But before I come up with a specific definition, let's see what the components of that operon is. So you will find in it is a gene here, another gene here. That means they are encoding sequences, coding sequences, 3 of them. These are called the Z, the Y, and the A. These are structural genes. When they are transcribed, and then translated, so transcribed and then translated, they will make 3 enzymes. Those enzymes are beta galactosidase, permease, and transacetylase. That's when these Z, Y, and A genes are transcribed and then translated. Now, those are enzymes. That's all what I'm going to say about them right now. They are enzymes needed to break down, which is catabolize the sugar lactose. And remember again, I'm not going to break down a sugar if the sugar is not present. So don't make them if there is no lactose. All right. What else I have in that lac operon? You have another sequence here and another sequence here. But they are not, look, they are not genes, meaning they are not transcribed and translated. It's just a DNA sequence. One of them is called the operator, and one of them is the promoter. What binds to the operator? The main enzyme of transcription, and I actually make it like bind it, which is RNA polymerase. That's the name of the enzyme, which is the main enzyme of transcription. Again, main enzyme of transcription. The enzyme that will do what? That will allow the transcription to take place. All right, going back again. Now, upstream of that, listen to the language. Upstream is actually this way, and this is downstream here. Upstream, downstream. There is another sequence right there, but notice it is not part of the lac operon. It is upstream of it. And that is called the I gene. But look, it's a coding sequence. So it does get transcribed and translated, and it's going to make a repressor protein. I'm going to make it look like this. This is repressor protein. So that I gene is a regulatory gene. The I gene is here, a regulatory gene. It does regulate. Now, it is transcribed and translated 24/7. 24/7. So it's always made. Anything that is made 24/7, then I guess it is not regulated. Like it's made. I mean, no one is stopping it from being made. So 24/7 being transcribed and translated. And the key is an enzyme. It's going to make sense once we finish everything. Right now, all of this is new. This repressor protein is made in its active form. And guess what happens when it is made in its active form? It's going to go right away and bind to the operator. So let's go back and do it here, right there. It's going to bind to the operator because it is made in its active form. So right now, you do not have lactose, right? I'm not showing you lactose. I don't see lactose. So the making of these enzymes, what enzymes? Those enzymes here will be a waste of time because these enzymes are needed to break down or catabolize the sugar lactose. What lactose? I don't have lactose. Then don't make them. So this is how it goes. I don't see lactose. The enzyme of transcription is RNA polymerase. This one here. But that enzyme must move down, remember, downstream to do what? To transcribe the Z, the Y, the A. Well, I can't. Why not? Because the repressor protein is in my way. Which is awesome, because you should not be moving downstream, because you should not be making those enzymes anyway, not right now. Okay, now, the second you add, look what will happen. The second you add lactose, let's agree that this is lactose here. Let's say I just added lactose here, lactose. Well, now I need to make those enzymes. Now I need to make them. So to make them, because I need to make them to break down lactose, what will happen is, see the repressor protein that is bound? The second you add lactose, that repressor protein will leave the operator and will bind to the lactose instead. So what will happen is, I'm going to get rid of it from here. Look, it's gone. And going back to the repressor protein in red, it's going to actually bind to the lactose instead. So when it is bound to the lactose, it is inactive. So now it's inactive repressor protein. And the RNA polymerase now will be able to read, to move downstream, and read Z, Y, and A and transcribe them, which is perfect, because now there is lactose and I need to break it down, so I need to make the enzymes. That's essentially what enzyme induction is. Let me repeat a few things or emphasize a few things. I said gene, I called it here, I said transcribed and translated 24/7. Then it's said to be unregulated, which makes it, that's the term used all the time, constitutive. If I say someone is studying constitutively, that means that someone is studying all the time, or all times. You drink coffee constitutively. Every time I see you, you have coffee with you. So it's really English as well, constitutive. So at all times being transcribed and translated 24/7, it's unregulated. There is no shut off and mechanism for it. It's just active all the time. Now, proteins that are made 24/7, again, proteins that are made 24/7, that means they are needed 24/7. They are said to be housekeeping proteins, because I need those proteins or enzyme 24/7. I need them 100% of the time. All right, so what did I say? I said that the repressor protein here in enzyme induction. Yeah, what about that repressor protein? Has a higher affinity, higher affinity for lactose than for the operator, right? Look again. The second I added lactose, first of all, the repressor protein was here, remember, was bound right there. Was bound here. But the second I added that lactose, it did leave that operator. It's no longer there. And it is now bound to the lactose itself. So lactose did what? So lactose inactivates the repressor protein. Did it not? Because I said it's when it's bound to the operator, it's active. When it's not bound, then it's inactive. All right, now the term, and I will do this still one more time to summarize it, because now everything should add up, but I still want to emphasize that concept. So once again, the term metabolism that includes the building and the breaking down. So it does include anabolism and catabolism. Anabolism is the making, the building, the biosynthesis. Biosynthesis, again, making, creating, building up. This one, the breakdown, which is the degradational pathways. From degrade, pouring down, again, degrade, to destroy, to fragment, to really put down, to humiliate. When you say to degrade, because you're breaking down, not building up. The two together, I said that's metabolism. Let's go back here. So now lactose is the inducer, isn't it? What does that mean? The presence of lactose induced the cell to make the enzymes that are needed to catabolize it, to break it down. So it is the inducer. Look at the diagram again. What made this repressor protein inactive? What inactivated that repressor protein? Lactose. And as a result, my RNA polymerase can now move downstream. And those enzymes are inducible. So those three enzymes, the beta galactosidase, permease, and transacetylase, they are inducible enzymes or inducible proteins. Again, inducible. They're not present 24/7. They are only present or only made when needed, when lactose is present. So that's the idea, again, of enzyme induction. So I'm going to do this one more time, but I want to do two different slides this time. With lactose, without lactose. So I'm going to try to do them next to one another like this way to compare. All right, so one more time, this is the lac, and I will define that term again, operon. I will do the same thing here. Lac. Here I'm gonna say no lactose. Here lactose is present. All right, the ZYNA, ZYA without, like, you know, these are the structural genes, same thing here. ZYA RNA, the structural genes. We can label them, that's okay. Here. Same thing. All right, here you also have operator, promoter, same, and promoter. They are labeled here, so I'm not gonna label them again. They are labeled right there. Here and here. And I said those are not genes. What should bind to them is, let's use black, RNA polymerase. RNA polymerase. Again, RNA polymerase, RNA polymerase. Now, when lactose is not present, again, no lactose, so the one to the left. Before even that, I forgot, you have the I gene here. I gene, same thing. I gene, and I said this is 24/7 transcribed and translated, and making that repressor protein in its active form. Same exact thing here, 24/7, 24/7, making that repressor protein. There is no difference whatsoever so far. Repressor protein in its active form. Now, no lactose, which is the one to the left, that repressor protein will go and bind right here. And I'm going to label that active repressor protein. All right, so when no lactose, which is the one to the left, the RNA polymerase attempting to move downstream will not be able to. Why not? Because the active repressor protein is in its way, which means the Z, Y, and A will not be transcribed, and of course will not be translated, which is amazing because I don't want to make them right now because they are only made to break down lactose. I don't see lactose. Now, in the illustration to the right, I'm going to add lactose. This is the lactose. Lactose. That repressor protein that you are staring at here will actually bind. Like maybe it was bound. I'm going to do it and erase it. Maybe it was bound here before you added lactose, because it was. Look at the diagram to the left. But the second you add the lactose, I'm going to leave this spot. I don't like this spot. I'm going to actually go and bind to lactose itself like this. And this is inactive repressor protein. So now RNA polymerase will seize the chance and will move downstream and will be transcribing Z, Y, and A. So the Z, Y, and A as a result here will get transcribed. I'm going to do this, transcribed, and then translated, and we'll make those three enzymes. those enzymes, beta-galactosidase, permease, transacetylase. And I will write them down again. We'll make, I'm going to actually extend it here. We'll make beta permease, trans. All right. Come on. Beta-galactosidase, permease, and transacetylase. And these enzymes are now made. And these enzymes needed to do what? To break down, which is the same as catabolize the sugar lactose, which is the inducer, as we said. That's it. You have to keep, you can see the sky is the limit in what you can ask about this. Now imagine when we are done with it and done with the enzyme repression, how many questions can be asked? So this is it. You need to know that when the repressor protein is bound to the operator, as it is here, then it's active. When it's not bound to the operator, as here, it's inactive. But it is made in its active form. It is made in its active form. It's synthesized in its active form. Now the term lac, well, first of all, the term operon. It is a DNA sequence. Before I finish the definition, well, you can't tell me it's not. Remember, I got it from where? From the chromosome. Here. Right there. Why this is not a DNA sequence? It has to be. It's a DNA sequence. It's from the bacterial chromosome, and the chromosome is made of DNA. All right, so going down, it is a DNA sequence that include related structural genes, such as Z, Y, and A. So includes related structural genes as well as regulatory sites. What are the regulatory sites? The promoter and operator. Because I said they are not genes, so I will emphasize again. Promoter and operator are not genes. They do not make proteins. They are sites to which other proteins bind. So the repressor protein binds to the operator, whereas the RNA polymerase binds to the promoter. So they are sites to which other proteins or enzymes bind. That's why we kept saying lactose is an inducer in this case. It did induce the cell to make the enzymes that are needed to catabolize it. And I still will say this, lactose lactose is the inducer. How come again? Because it induces, or it induced, the cell to make the enzymes needed to break it down, to break it down, to catabolize it, catabolize it. And how? So by doing what? By inactivating, the repressor protein. And when I say by inactivating the repressor protein, thus the RNA polymerase can now move downstream and transcribe Z, Y, and A. Z, Y, and A. That's what enzyme induction is. So enzymes that are made only when needed are said to be, remember, inducible. We'll use that term again. Inducible enzymes. Inducible enzymes or inducible proteins. All right. But there is more to the story. There is one more component that we must address as well, which is they discovered that if you incubate bacteria with glucose here, so this is number of bacteria, over time, and they are incubated them with glucose. That's what happened. So the sugar used here is glucose. Bear with me. So over time, they are increasing exponentially in number. Fine. So they are enjoying that glucose. Now, when they incubated them with lactose, I'm going to use red for lactose, two things were different. One, there was a lag in their utilization of that sugar, and they were not enjoying it to the same rate. The slope was not as steep. So this is the lactose. So there was a lag right there. Like, why did they not increase in number right away? Whereas they increase in number right away with the glucose. So the first conclusion, just based on that alone, we already know why it took them some time to metabolize lactose. Well, because the enzymes needed to break down lactose were not present, so we needed the time to make them. So that is the lag time, which is simply the enzyme induction time. Enzyme induction time, which is the time needed to make beta-galactosidase, permease, and transacetylase. I need time to make them. Yes, you gave me lactose, but I wasn't ready for it. So give me time to make the enzymes, because those enzymes are what again? Inducible. Okay, so, but fine, we knew that. So, but what's up with glucose? Why did they use it right away? That's a conclusion. then the enzymes needed to metabolize the sugar glucose seem to be housekeeping, seem to be present 24/7, because I did not hesitate to use it. So the other conclusion is the enzymes, this wasn't a conclusion so far, this is a fact that we knew. But now the enzymes needed to catabolize the sugar glucose are housekeeping, and another term, remember, is this constitutive. And that's why I did not, there was no lag time, there was no delay. It was instant. And I'm gonna add, whereas, this is not a conclusion, this is a fact, whereas the enzymes needed to catabolize the sugar lactose, as we already know, are inducible. I said this is a fact we knew, but this is the conclusion right there. That is the conclusion from this diagram. But that took care of what? That took care of why they did not hesitate to use the glucose, because they said the enzymes were present, will always be present, whether glucose is there or not, based on the observation. But the other one, like why they were enjoying the glucose more than the lactose. Well, remember, the glucose is a monosaccharide. It's easier to break it down. That's why they were increasing in number at a faster rate, because glucose is a monosaccharide, whereas lactose is a disaccharide. It's going to take more to break a disaccharide than to break a monosaccharide. So this is ready for like to use it for energy. I said it's easier to use it for energy than to use a disaccharide. It's a bigger molecule. The lactose is a bigger molecule, so you need more time to digest it. All right, but keep in mind, don't forget this, that the enzymes needed to catabolize the sugar glucose are housekeeping, constitutive, present 24/7. But now they did this. They said, okay, fine, let's incubate the bacteria with both sugars. And that was another observation. So here, bacteria, again, number of bacteria. And this one is over time. And look what happened. Let's use black. They incubated the bacteria with both glucose and lactose. Again, glucose and lactose. That's what happened. That looks like what? That looks like this line. It seems like they are using glucose and neglecting or ignoring the lactose. But when glucose ran out, now they needed time to make the enzymes. And then they started using the lactose at a slower rate. So now they said, okay, well, something is, for some odd reason, when you incubate the bacteria with both glucose and lactose, they ignore lactose and use glucose first. When they run out of glucose, I guess now they are forced to use the second sugar. So one more time, at this point, this is when glucose ran out. Glucose ran out, right? And this here, back again, I'm going to use the same language I used seconds ago, the lag time, the enzyme induction time, lag time, the time needed to make the enzymes that are needed to metabolize lactose. But as long as glucose was present, for some reason, I did not care for that lactose. We need to know why at the DNA level. This, what you are staring at right now, that's called glucose effect, which is basically why glucose will affect the metabolism of all other sugars. Another name for it is catabolite repression. Please, please, please do not confuse catabolite repression with enzyme repression. They are two different things. I did not even get to enzyme repression. I'm still looking at enzyme induction. So here, catabolite repression, I will define it, but has nothing to do with enzyme repression. Nothing whatsoever. Do not confuse the two. Now, what's catabolite repression? Catabolite repression, from the name, or glucose effect. Look, in the presence of glucose, hence the name glucose effect, the catabolism of any other catabolite and a catabolite is a source of energy is inhibited. One more time. In the presence of glucose, the catabolism, the breaking down of any other catabolite, any other source of energy is inhibited, because it seems like glucose is the preferred source of energy. This is your favorite dish. I mean, I'm giving you, 7-course meal. I mean, the different things you are eating, lunch or dinner. I mean, which one you will eat the most of? I guess the one you like the most, and you might ignore all the other dishes. This is not really any different, but I need to know how cells will know how to do this at the DNA level. How will they know to use glucose and ignore, let's say, lactose or whatever other sugar we have present? So in the presence of glucose, we really don't care about what other sugars you are adding, because they are not going to use it. As long as glucose is present, they will ignore all the other sugars. The two reasons for that, because we said the enzymes needed to metabolize glucose. What about them? They are housekeeping, they are constitutive, they are present 24/7. And glucose is very easy to digest through glycolysis. Remember, that's why glycolysis is a big deal. Through cellular respiration, it's a monosaccharide. It's a simple sugar. Lactose is not a simple sugar. All right, now, the reason for that is going back to this diagram here. Look at the DNA level. Going back to which one here? What I said earlier, look to the one on the right when lactose is present. I said as long as, now look, let's look on the back, on the left. I said as long as this guy here, the repressor protein, is not bound. Now going back to the right, I said RNA polymerase will be able to move downstream. Well, that's not precisely accurate. Why? Because I need to push RNA polymerase this way. So there is another complex, I will use yellow for it, that must actually bind here. And that is the one that will kickstart the RNA polymerase. Now, what is the name of that yellow, this guy here, molecule that must kickstart the downstream movement of RNA polymerase? The name of that yellow guy here, that yellow that I had above, is called cyclic adenosine monophosphate catabolite activator protein complex. Again, cyclic adenosine monophosphate catabolite activator protein complex. This is the molecule that must do what here? This guy must bind upstream of the promoter. Before I continue, look, I must bind where? Isn't that, we said, where is the diagram earlier? Okay, I said when you are going here, when you are going this way, that's upstream. When you are going this way, that's downstream. So in this diagram, isn't that RNA polymerase right there? This one is RNA polymerase. I am bound upstream of it. And RNA polymerase is bound where? Is bound to the operator. I'm sorry, to the promoter. Here. Promoter. Where are we? So upstream of the promoter and kick start the downstream movement of RNA polymerase. So one more time, that complex must bind upstream of the promoter and kickstart the downstream movement of RNA polymerase. For RNA polymerase to start moving this way to get to the Z and the Y and the A, I need to be kicked. Who's gonna kick me? This guy here. What is the name of that guy? This here. Has to kick it. And this is the RNA polymerase. So now, going back to catabolite repression, I'm gonna say the molecular mechanism of catabolite repression, which is the same as glucose effect. We said when glucose is there, I'm not going to be using lactose. Let me show you why. So glucose is very high. So here, very high glucose. All right, when glucose is very high, that means you have tons of adenosine triphosphate. Isn't that, they are correlated, right? High glucose, high ATP. But when you have high ATP, you can't have triphosphate and monophosphate at the same time. So this will be very low adenosine monophosphate. When you have low adenosine monophosphate, then the cyclic adenosine monophosphate is also low because it's just the close form of adenosine monophosphate. Now, when the cyclic adenosine monophosphate is low, then the cyclic adenosine monophosphate catabolite activator protein may not even form. Well, because I barely have enough cyclic adenosine monophosphate. Or it may form, but at very low concentration to bind and kick start the downstream movement of what? Of RNA polymerase. What does that mean? That means Z, Y, and A are not transcribed. What does that mean again? That the enzymes needed to catabolize lactose are not made, which is exactly what we said. Because I don't need to make them. Why should I make them if, look, if I have high glucose, then I'm not going to be using lactose. That is the molecular mechanism. So glucose is very high. That means adenosine triphosphate is very high. But when adenosine triphosphate is very high, the monophosphate is low, which means the cyclic form of it is low. When the cyclic form of it is low, so don't expect the cyclic adenosine monophosphate catabolite activator protein to form. And even if it does form, it's not going to form at high concentration or enough concentration to actually bind and do the kicking. So you will not be transcribing ZYNA, and you only need to transcribe ZYNA if you need to metabolize lactose. But you're not really in need to metabolize lactose right now because there is glucose, which is easier to metabolize. It's a monosaccharide. That's one. Two, the enzymes needed to break it down are already present 24/7. That's the mechanism. Now, if I want to reverse everything, and we can, so let's say now glucose is running out. Remember, look here, I don't have any more glucose at this point. So I'm freaking out. Okay, so reverse everything I just said. So glucose is very low, barely have any. Done. I'm running out. That means adenosine triphosphate is very low. That means adenosine monophosphate is very high. Remember, if this is low, this is high. That means the cyclic adenosine monophosphate is high. That means the cyclic adenosine monophosphate catabolite activator protein complex will form, which means will bind upstream of the promoter and will ignite or kick start the downstream movement of RNA polymerase, which means Z, Y, and A are transcribed, which means the enzymes needed to break down lactose are now made. That's why I'm going to be using the lactose, because, well, I don't have any glucose left. All right. for enzyme repression. Here, what was my analogy? I said a bacteria cannot live without tryptophan. They have to have tryptophan present 24/7. No tryptophan, we have a problem. So they have to make tryptophan themselves. So the second you offer them tryptophan, you will repress them from making the enzymes that are needed to make the tryptophan. Think of it like a job. You make whatever, let's say you make, what is it? Let's say you make $100 a day. Well, and you have to make $100 a day. Well, if someone is giving you the $100 without going to work, well, I guess that will repress you from caring about your job. The fact that someone is giving you the money, I don't think you will care for that job. But you're not complaining. If no one is giving you the money, you go to, you do your shift, whatever, and you make that money that you have to make for that day. Once again, the second someone is giving you the money, that will repress you from caring about that job. You become lazy. And this is not any different. So now the only difference in the cascade is this. So you have the trip, same thing, see trip for the amino acid tryptophan, operon, and you still by the way have the two regulatory sequences, and these are the operator and the same. The operator is this one, and the promoter is this one here. So that's not any different. But now instead of Z, Y, and A, there are actually 5 of them. 1, 2, 3, 4, 5. And I'm not going to give you their names. They are called A, B, C, D, E without the specific names, but they actually start with the A here. So A, B, C, D, and E. These are the structural genes. And once they are transcribed and translated, they are going to make enzymes here, enzymes A through E, and these are enzymes needed to do what? To make the amino acid tryptophan, not to break it down here, to biosynthesize, which is to make the amino acid tryptophan. So to make it. And the same thing, upstream of that, you had that I gene, which is constitutive, 24/7 made, transcribed and translated, making that repressor protein. And let me do one more thing, which is RNA polymerase is bound here. So I don't see any difference yet. RNA polymerase. Guess what, guys? The only difference, please pay attention. The only difference, you do not want to miss, to me, the easiest questions, because I said they are conceptual. The only difference is the repressor protein in enzyme repression is made in its inactive form. Here, made in its inactive form, not active form, inactive form, which means cannot bind. So this is how it's going to look like. That's it. Do you see tryptophan? No tryptophan. So am I complaining? I'm not complaining, meaning what? The RNA polymerase will be able to move downstream and read the A, B, C, D, and E, transcribe, and then the translation will follow and make those enzymes that are needed to do what? To make the amino acid tryptophan. This is how it's going to look like when tryptophan is absent, because I have to make it. I said, I have to make money. If you're not giving me money, I have to make money. I cannot live without money. All right. Here, it's the second you add tryptophan. Let me make tryptophan green and triangle this time. These are the tryptophan. So now I'm giving you money. I'm not going to care about your job because I'm giving you the money. So these are a tryptophan. Tryptophan will activate the repressor protein, and the two will bind to the operator. So that's what will happen. See, I added tryptophan. The second tryptophan is added. The repressor protein in red here, repressor protein will become active. So it's going to bind. But what is activating that repressor protein? Going back to my green, that tryptophan is bound to it. All right, so tryptophan right here activates the repressor protein. And thus, the repressor protein tryptophan complex binds to the operator, preventing the downstream movement of RNA polymerase. And why will I don't remember if that is being prevented from moving downstream. I'm not gonna make those enzymes that are needed to do what? To biosynthesize the amino acid tryptophan. And why do you care to biosynthesize it if you are giving it to the cell? Why would you care to make money if I'm giving you the money you are making daily? Like I'm just giving it to you. So I guess I said you're not gonna care to make the money yourself. So here tryptophan, I said it's activating the repressor protein. Now remember what we called lactose? We called it the inducer. Tryptophan is said to be the co-repressor because it's activating the repressor. Because, yeah, so tryptophan is called the co-repressor because it activates the repressor protein. It's making the repressor act as such by activating it. So the major difference in enzyme repression, the repressor protein is made in an inactive form, which means cannot bind, and it has to be cannot bind to the operator. Tryptophan will activate it. The other way around. In enzyme induction, the repressor protein is made in its active form. And the presence of lactose will inactivate that. Whereas the presence of tryptophan will inactivate. So be careful with activate and inactivate. Yeah. That's huge. Let's see here. One last thing about this slide. I mentioned that cyclic adenosine, whatever, monophosphate, catabolite, activator protein, I said it's needed to kickstart the downstream movement of RNA polymerase. But that's only needed in enzyme induction. So I'm going to emphasize that. This guy, cyclic adenosine, monophosphate, catabolite, activator, protein complex is only needed in enzyme induction, meaning it has nothing to do with enzyme repression. So I'm not going to come here and say, oh, guys, you have that complex. No, that's not true. I'm going to, here, see that one? That's garbage. I'm going to get rid of it right now. There is no such thing needed here because we are talking about enzyme repression. So let me get rid of it. It doesn't exist. You only need it for enzyme induction. Enzyme induction. Did we? Here. Sorry. It was right there. All right, I will stop this video at this point.